going to talk about this paper. So I met the author when I went for training down at the University of Maryland, Baltimore campus. So she was the EM facilitator. So she imaged all of the phage, and she was the one that imaged this, which she then became first author. So this is like, referred to as the vampire phage. So what you see here is you have two phages. You have your helper phage, and then you have the little satellite phage. All right, so I know this paper, it's a cool paper, but I forgot how dense it gets with the genomics. So we are going to go over that and make sure you guys are comfortable with what they were trying to tell you about in this paper. Okay, so table 1. So here they're just looking at two helpers and two satellites. So Mind Flayer is the helper, and its satellite is Mini Flayer. The other helper is Mulch Mansion, and the satellite is Mulch Room. So here are the statistics from one of their genome runs. So mapped reads is referring to the abundance that they had in that sample. And you can see that both of the satellites were much more abundant compared to their helper. So they are extremely abundant in every sample. When you compare the genome lengths, the two satellites are genome-wise much smaller, whereas the helpers, 130,000 length in base pairs. Percent GC, so they definitely talk quite a bit about this. So this, they were surprised with this because it's under, it's a low GC content genome, which they were not expecting. And what's even more exciting, I guess, for them was when you compare the GC richness. So does everyone remember what ATGC is, the bases of DNA? Okay, this is important because you can get some really interesting biology if you have an AT-rich genome versus a GC-rich genome, especially when you get to extreme animals that can, withstand freezing, like some fish. They typically are AT-rich and not GC-rich. So it's really, really interesting. So they keep track of this. But then when you compare this to the host that they infect, the host is 70% GC rich, but both the helper and the satellites are AT rich. Okay, so they have a low GC richness. Yeah.
The host is the cell that they...
Yes. Yeah. So I think this was a Streptomyces bacterium. Okay. And then they just give a list of the genes that code for proteins. And then the transfer RNA genes. So remember, in our genome as well, we have genes that make our mRNA. We have genes that make our tRNA molecules, our rRNA molecules. So that is built into our genome. Now, when you look at a codon, so messenger RNA, when it gets translated, it is translated by triplet codons, right? Everyone good with that? So AUG is for methionine for every protein we know of. So knowing that, we also know that for every transfer RNA, it has its own protein to charge it. So it will go, when it needs to be attached to the correct amino acid, it will go to this enzyme, and then the correct amino acid would be attached to it. So when you look at a codon chart, there are multiple options. Like for serine, there are 6 different codons that will put serine on a transfer RNA. But there's 6 different transfer RNAs because they're all specific to their anticodon, right? You okay still? Okay. Now, out of those 6 transfer RNAs that will be for serine, there is a bias. So not all of them are used equally. So in serine's case, there is one that's used 80% of the time. So there's a codon bias that I think it's ATG, that one is the one that is, you know, most prevalent in a genome or a messenger RNA. So this is what they're about to get into with this. So they looked at the transfer RNA genes. Again, they're, you know, we have 64 different codons. There are 64 different transfer RNA genes for each of those codons. The helpers have these tRNA genes in their genome. The satellites do not. So what does that mean? If the satellites don't have the tRNA gene, they can't make tRNA, right? So what does that mean for satellite and helper phages? Yeah, Thomas.
Helpers like tRNAs have to like transcribe or translate the satellite.
Exactly. So these satellites completely rely on their helper phage to do all of the translation for them. And then they were kind of getting at some of the really interesting helper satellite biology that they know of. Sometimes the helper is, you know, trying to infect a bacteria and lyse it and get rid of it and all that stuff. But if there's some of the satellites try and inhibit that. So it's almost like pro-host. Others, it's the reverse, where they work with their helper to lyse and take over the bacteria. So, you know, that's going to be a whole interesting behavior almost, even though these aren't alive, you know, aspect to that they're going to be looking into. Okay. So here is some of their genomics that they did. So at the top, they're comparing the two helper genomes, so MindFlayer and Mulch-Mansion, and you can see the color-coded functional genomics where they appear within the genome, and then the gray slashes are just for similarity. So the darker, the more similar, the lighter gray, the less similar. So when you kind of look through the paper reports that between the two helpers, there's 62% similarity between the two genomes. So that's pretty good. For the two satellites, so MiniFlayer and Mulch-Room, you know, they did the same type of experiment. Again, it's a much smaller genome, but you can see there's definitely not as much similarity, okay? So that's kind of what you get out of this. Then here are their TEMs. So for the top one, that is Mulch-Room. So that is the helper. No, Mulch-Mansion is the helper. That is the satellite, shown in A, I guess. This one's MindFlayer, which is the helper. And then we have MiniFlayer, so that's the satellite. So these two go together. Then D is MiniFlayer, which is, okay. So this one is on here because it's showing the attachment of the satellite to the helper tail. So the satellite does not attach to the host. It doesn't attach to the capsid of the helper. It attaches to the tail protein of the helper. So it will inject its DNA into the tail, and then its DNA will make it into the host through the helper tail. Okay? All right. Then they showed, you know, with they call these bite marks, but they can tell that a satellite had already been attached because it leaves a little bit of tail fiber there of the satellite. And so most likely when it was attached, it injected its DNA into the tail and then it infected a host, but now it has fallen off. So, and then finally, the last picture, so this is showing the whole association. So here is the host bacteria. And again, I think it's Streptomyces. We have the helper attached to it, and then the satellite attached to the tail of the helper, okay? So those images are just kind of showing you how it all fits together. All right, then they did a full genomic phylogeny. So they have a lot of information on here. They have highlighted the two satellites. So satellites are also indicated by stars. So the mini flare and mulch room are shown up there. These are all BE cluster phage. At least the ones that they have sequences for. And then the two helpers are also highlighted in yellow over here. Then when you go from working your way out, so it's showing the relationships. The bootstrap is indicated by the size of this bubble. So the larger, the more significant, a higher bootstrap number. And then when you get out to this is just the names. But then the first green row of bubbles or moons is showing the GC, it's showing if it's temperate or virulent. So you can see when you kind of scan through here, most of the BE clusters are temperate. So they were really excited when they saw this, like even the helper, they have, a lytic pathway. So it's kind of interesting. Now, the mulch room, it is a lysogenic helper, lysogenic satellite. But when it hooks up with its helper, those lysogenic genes can kind of help it integrate into the host genome, is what they're thinking. So you've got your temperate versus virulent conditions. Then your last row is the GC content or richness. So if it's totally purple, which this one is almost all purple, that is a GC rich genome. So that's a lot of information on one figure. So that's just kind of telling you what they know about the BE cluster, how they're related, if they're virulent or temperate. Okay. All right, so this figure, so figure 4. So remembering what we just talked about with transfer RNA and the codon bias, what did you guys get from this figure? What did you make of it? So first of all, what are the orange bars? Yeah. Okay, so phage transfer RNA. What are the blue bars? Host tRNA. And then they have the two satellites kind of just shoved because we know that they don't have tRNA genes, but they're doing it as a comparison to their helper, knowing that they're going to use the helper's tRNA genes to make their, in this case, I think it was the major capsid protein. All right, so what else is this kind of telling us? So we're comparing phage DNA to host DNA. So our bacteria and their transfer RNA genes and usage, do they map well together when you compare the phage to the host? I mean, they're not horribly different, but there's quite a bit of difference when, we're looking at about 0.7 adaptation of the tRNA compared to more like 0.5. Okay, so they are using, their transfer RNA, differently, okay? Again, this is the BE cluster that is represented here. All right, so let's talk a little bit more about codon bias. So there's lots of different codon charts. I'm sure you guys have had to use these before. But there's definitely a movement to starting to look at those differences because what is a silent mutation?
The one that doesn't change the codon or the amino acid gets that.
Okay, so what position does that happen in?
The third.
The third of the triplet codon. So that is the wobble effect. I'm sure you've heard before. So knowing that, yes, you can have 6 different codons and get a serine put in, but that's also 6 different transfer RNAs, 6 different enzymes to charge that transfer RNA. And particularly for serine, 80% of them use the one type. All right, so what you're seeing here is just a codon chart, but then it's got 5 different organisms, all the way from humans to yeast and bacteria is up there somewhere. And what they're showing is when you look at just a sample of some of the codons, so for isoleucine, you've got these 3 options. Okay, so that wobble effect, that gives you a silent mutation. So if you switch this to, I don't know, anything else, like AUA, you're still going to get isoleucine, okay? But that can still affect the protein translation because you're calling for a different tRNA to bring isoleucine. So that's where the bias really comes in. And so this chart is trying to look, for lysine, for all 5 of these organisms, they all prefer AAG. Even though this will bring them lysine, it's not selected for as much. Okay, so they're just starting to look at those silent mutations, which we really thought were benign. There could be an effect to translation processes. And remember, these are bacteria. So what is one huge difference between prokaryotes and eukaryotes? Grant?
Sexual selection.
Well, definitely that too.
DNA is like wrapped up in histones and chromosomes for eukaryotes and prokaryotes. It's kind of loose.
Yeah, so the DNA is protected and, wrapped around histones, but also what's that other layer of protection for eukaryotes? It's in the nucleus. Bacteria, it's all in the same location. So you can have transcription and translation going on at the same time. So if you're a bacteria and let's say you're in a human gut and the human gut that you're in, let's say you're malnourished. Let's say you're not eating a balanced diet. And now all of a sudden you may not have all of the amino acids, in abundance to make constant translation for this bacterial organism. So if you have a stall where, the ribosome's chugging down the messenger RNA, doing its translation thing, but if it is sitting on a codon waiting for a very low abundant transfer RNA and you're malnourished, if it doesn't come pretty soon, it gets knocked off and translation stops. So this codon bias is something that, for bacteria, this could be, make or break for translation. Efficiency is always number 1. And if they don't match, especially satellite to helper, really well, or even to their host, if they're going to take over the host machinery to do their translation, they really need to kind of be in sync because you can get, complete shutdown of translation, okay, if you have silent mutations that aren't preferred, okay? So that's a big part of what this paper is starting to look at with a very simple transfer RNA adaptive index summary, which you can pull up through GenBank and some of the other sites. So if this interests you, here is a link where you can start to look at some of the work that's been done on codon usage frequencies. So it's not specifically tRNA, but it's, anything you can go on and they have at least 10 organisms that you can select. And then you also have to know the letter code for the amino acids. Okay, so you probably know the abbreviation like LIS is lysine, but you also have to know that when you were looking at genome data, lysine is represented by a K. Okay, so here's just a little cheat sheet so that if you open this and play around on it, you're just going to see a ton of letters. Each letter represents one of our 20 amino acids that are used in proteins. Knowing we have more amino acids than 20, just not all of them, only 20 are used in proteins.